Yep, let's do it.
Great. So thank you so much for having me here today, and of course, for the lovely introduction. So as part of your topic series on cryopreservation, I'm going to talk to you about a high sub-zero cryopreservation approach that we call partial freezing. The idea behind partial freezing is to promote a thermodynamically stable frozen state while maintaining a sufficient unfrozen fraction to really limit ice damage, but also excessive dehydration. So before we jump into some of the theory of partial freezing, I'm actually going to start with a brief introduction of the importance of cryopreservation, which you've probably heard already. And then I'm going to focus on one application in particular, which is organ transplant, and then we'll sort of come back and do a little bit more of a deep dive on the theory behind partial freezing and how we're using it in the lab. So in an era of medicine where therapeutics are consisting more and more of living entities, advances in cryopreservation have really become an immediate necessity. The ability to disseminate living systems really has a significant role to play in many broad areas of science, engineering, and of course, medicine. So for example, if we think about the advent of new cell therapies, CAR T cells, for example, or maybe the application of organ on a chip or other microphysiological systems that are used as aids in drug discovery or as a means to replace damaged tissue. These are all live therapeutics, but also research tools that require efficient cryopreservation in order to get into the hands of the people who need them, including the patients. We can also start to think about more complex systems, such as vertebrate model systems, and you'll see a little bit of that in the talk today. So this can include things like zebrafish or fruit flies. So if you think about a fish facility, for example, it can spend hundreds of thousands of dollars to keep the facilities running, especially now when there's this enormous toolkit of transgenic lines that continues to increase. This storage problem really becomes extremely problematic. There are also environmental issues that are very much on the mind of everyone right now. We have a changing Earth, and cryopreservation is one tool that we can use to help protect biodiversity. We have to think about how we're going to feed a growing population and perhaps even one day send humans for long-term space travel. But finally, one of the applications that really guides my research, and what I'm going to talk to you about for a couple of slides, is the ability to preserve transplantable organs. So why is this so important? If we look at the landscape in organ transplantation, there's this massive discrepancy between what the potential of organ transplant could be versus what it is right now. It's estimated that approximately 35% of all annual US deaths could be prevented by organ transplantation. That's if you remove all barriers on transplantation. In other words, if anyone needed an organ, it was directly available. This could translate into 1 million lives saved every year in the US alone. So these are significant numbers that we're talking about. However, if you look at what's currently occurring, we only have about 40,000 transplants that were performed in the US in 2020. So this is really despite amazing and impressive surgical advances that have really enabled this field and enabled organ transplants to be routine life-saving procedures with really impressive median survival times. So if you think about this, this is actually a problem that's contrary to many other illnesses. For patients with end-stage organ failure, there's actually a life-saving technology out there, but it's often inaccessible. So this is an inaccessible cure. Of course, there are complex factors that influence this discrepancy between supply and demand, and I'm only going to talk about 3 of them here. And then again, we'll just focus mainly on that preservation storage duration. So preservation duration really creates this logistical nightmare. We have very short preservation durations for whole organs. For example, a heart can only live outside the body for about 3 hours, a liver about 9. These can vary slightly from transplant center to transplant center. These are the MGH numbers, but the point is that these organs can only survive a couple of hours. The second problem we have is we have an extension or expansion of the donor pool. And basically what we're trying to do here is revive or resuscitate organs that are marginally injured. So these are organs that have currently not met the standard for transplantation, but they're almost there. And if we could have a means to revive those, we could significantly expand the donor pool. The third, which is intimately related to the other two, is the ability to have an objective and quantifiable viability metric that would allow us to know for sure if an organ can be transplanted. This would allow us to move away from that extremely risk-averse scenario we have at most transplant centers, and really help actually improve the number of organs that are discarded. So unfortunately, I'm not going to have time to go into all of these. And now let's really focus on that preservation duration and what we're doing to help overcome it. But there are many ways that preservation could impact organ transplantation. For us here in Boston, we have a very short window of time, and this really limits the geographical area in which we can serve. But if you can imagine for a moment that we could expand that from hours to multiple days, that could really enable national or even perhaps international sharing. And I have a feeling Corcutt might have even shown you the exact same slide. But what does this really mean? So one of the things that this would allow us to do is to convert from emergency to elective surgeries. This is critical because not only would this increase recipient outcomes, but could also increase the quality of life of transplant surgeons who don't have to wake up in the middle of the night to run in to do a transplant. It would reduce the economic burden, so reduce the cost of transplantation, for example, by eliminating the need to charter private jets without notice. It actually has been shown to directly reduce the organ discard rate. So there are some organs that are discarded simply because they can't make it to their recipient on time. The other way in which this could occur is that it would also impact organs for research. So of course, not all of those organs that are discarded because they can't make it to their recipient, but some of those could actually be used to disseminate for research purposes. I know for sure I could significantly benefit by a tool such as that, or more access to organs for research. That's some of the ways. There are also new breakthroughs that are occurring in organ transplant that would also be significantly effective for preservation. For example, we have immune tolerance protocols that need more time. That's simply not feasible with current preservation modalities. We have the types of transplants that are changing and becoming more complex. They require matching not only by immunological factors, but aesthetic ones as well, such as a hand or a face. We also have complementary fields such as tissue engineering and also regenerative medicine, which are really aiming to achieve much longer term breakthroughs in the development of synthetic tissues. And in order for that to be feasible, we need to have some sort of metric or some means to have a central banking facility and then dissemination from those central facilities. So this is why I like to think about preservation, especially in the space of organ and tissues for transplantation, as an enabling technology. It's needed now to help some of these logistical constraints that I mentioned, but it's also needed in the long term. So now I'm going to bring you a little bit into the weeds on some of the different approaches we have in literature, and then I'll focus in on some of the high sub-zero approaches like partial freezing. But I'm probably going to spend a lot of time on this slide, and it's really an important slide. If you look at what we have on the x-axis, on the y-axis, we have temperature. And temperature directly correlates to the storage time. So in other words, as you decrease your storage temperature, presumably you should be able to increase your storage duration. So if you think about hypothermic storage, which is the clinical means to store whole organs currently, it's around 4 degrees Celsius, you can achieve a couple of hours of storage duration of complex organs, like we've already mentioned before. As you start to push down in temperature into, for example, supercooling, partial freezing, and even further into vitrification, what happens is you decrease temperature, so therefore you decrease the overall metabolism of the system. And what that also means is that you decrease the need for nutrients, for oxygen, and presumably can increase that cold ischemic time, or that time that the organs can survive without oxygen and nutrients for much longer. So that was the idea behind supercooling, the ability to go below the freezing point, push down temperature, decrease metabolism, but stay within a liquid state. So supercooling is below the freezing point, but still maintenance of the preservation state in that liquid state. The second approach, partial freezing, I'll talk a little bit more about how that's different from supercooling and why we justify it. But basically here we push down into deeper storage temperatures in the range of -15, maybe as cold as -30, we'll see. And again, enabling colder storages, temperatures, deeper metabolic depression, and hence longer preservation times. And these two approaches are what we call high sub-zero approaches. On the other extreme, of course, we have vitrification. I'm sure you've already talked about that. So this is transformation into a glassy state, but storage at really cold temperatures, negative 140 degrees Celsius and below. And this could presumably allow for true banking of organs, so banking on the order of years. However, there are many challenges in the landscape currently for vitrification, especially as it applies to whole human organs. So we kind of view these high sub-zero approaches as an intermediate between hyperthermic clinical storage and vitrification, that we could perhaps achieve these, make this feasible in the shorter term, but still have a significant impact in the field of organ transplantation. The other approach that I want to mention briefly is up in the higher realm, so more closer to the physiology of the organ. There's two approaches: normothermic, which is at normal physiological temperatures of 37 degrees, and subnormothermic, which is around room temperature of 21 degrees Celsius. So this is different from the others, which aim to achieve metabolic rate depression. These ones actually achieve to have metabolic support. So in other words, these are typically active preservation modalities. In other words, they're typically matched with the machine perfusion technology as a means to deliver nutrients, oxygen to the organ, and sort of mimic the blood in a sense to keep the organ alive during transport and during preservation. The other important thing that I want to highlight here is that I actually don't view these as competitive technologies. I think there's actually space for many of these, and we can mix and match them in strategic ways so that we can gain the benefits of some and get rid of the disadvantages of the other, because really all of them have some advantages and some of them, and all of them also have disadvantages as well. The other thing that I want to highlight is that there are actually organisms in nature that display examples of each of these approaches. So for example, we have primates that can go into suspended animation at normothermic and subnormothermic temperatures. We have squirrels that can supercool, and we have freeze-tolerant organisms, the Rana sylvatica, which is going to be the highlight of this talk, that can freeze completely solid, and even rare instances in nature of organisms that can vitrify. So now focusing in on supercooling and partial freezing, I'm going to breeze through quickly the concepts of supercooling, because I know you already heard that from Dr. Uygun, and then we'll jump into partial freezing. So supercooling preservation, again, one of these high sub-zero approaches. The purpose is to preserve in an ice-free preservation modality. So again, it's below the freezing point, but we don't want to initiate ice. The thinking behind this is, again, greater metabolic rate depression, but not having to deal with phase changes of water. There was a couple of really important parameters that made this successful. This is the original team that developed the protocol. It was originally developed in rodents and was able to show a 3-fold longer extension of preservation duration as compared to clinical standards. And there were 3 important prongs, or 3 important pillars of the protocol. The first is it had to be matched with machine perfusion. So this is the perfect example that I was trying to highlight from the previous slide, that really these are not competing technologies and they can be mixed and matched in strategic ways. So machine perfusion was used as a means to load and unload important cryoprotectant agents, but also to revive and assess the organ after. It also included the static supercooling preservation step, which stored at -6 degrees Celsius and achieved that deeper metabolic stasis. And then there was also important cryoprotective agents that were introduced prior to cryopreservation to be able to stabilize the supercooling state, but also ensure that cells, mammalian cells that can't normally survive that temperature can survive. One of the challenges in supercooling, however, is it's thermodynamically unstable, meaning that there's many challenges in translation, because as you increase the volume, you're going to also increase the risk of accidental ice formation. And accidental ice formation is very critical. That could be much more damaging than initiating ice at higher temperatures. So when we wanted to translate this to much larger systems, we had to really overcome that, and we used multiple approaches. We kind of tricked the system a little bit. So instead of using -6 degrees Celsius, we raised the temperature to -4. We also added more preservation agents into the solution to try and stabilize that supercooled state. So in other words, we decreased the freezing point of the solution. also use machine perfusion, and with all of these things together, we were able to scale this up to a whole human organ. However, the problem with supercooling is that it's going to be inherently limited in metabolic stasis. In other words, what I'm trying to say is that every time we want to decrease temperature, we're going to have to battle this risk of accidental ice formation. So if we really want to achieve much longer storage durations, we have to accept that ice is going to be there. And instead of fighting against it, if we initiate it in a controlled and safe way, then we can actually enable us to go down to much deeper storage temperatures. So I'm just going to play a quick video. This is adapted from a TV documentary. This is my PhD supervisor, Dr. Ken Story. He's going to show you the champion of what we call freeze tolerance in nature. This is Rana sylvatica. Please let me know if you can hear the audio.
These are the wood frogs that were caught last night.
The freezer is set at 8 degrees below freezing, roughly the temperature of the ground in winter. In a matter of hours, they'll be hard as rocks.
We have fully frozen Rana sylvatica wood frogs. They're probably 50–60% ice now, and their hearts are stopped, their brains are flatlined, their limbs can't be moved, they would be broken off. Yet they're still alive.
But the most amazing thing is the fact that the wood frog comes back to life.
So that's Rana sylvatica, the wood frog. So as Dr. Story mentioned, for all intents and purposes, this is an organism that is completely dead by our clinical standards. So there's no breathing, no heartbeat, no circulation, no nerve signals. It's frozen completely solid. 65% of its water is frozen as ice. However, after a certain period, usually on the order of a couple of weeks, this organism can thaw, completely reanimate, and carry on with its life as usual. So really what our aim is to be able to try to translate this mechanism, this freeze tolerance mechanism, and translate those into a protocol that can be used with mammalian cells that cannot survive this naturally. So how do we approach that? There are a couple of ways in which we approach this. I'll talk about two of those major approaches, and I'm going to jump into some of the data. One of the things that we use are a couple of biomimetic agents. Two of those that I'm going to talk about are a glucose analog that we call 3-OMG. One of the main crop protectants that a wood frog uses is glucose. It synthesizes massive amounts of glucose on the order of about 400 millimolar. However, the problem with that amount of glucose when introduced to mammalian cells that are not typically freeze tolerant, that can be highly toxic. When you have that amount of glucose, what happens is glycolytic enzymes start to break down the glucose and they produce lactate. That can cause drastic pH changes, apoptosis. The cell does not like that type of environment. So we have to trick the system a little bit. We use a glucose analog. It's basically glucose in structure. We basically throw a methyl group on there. And what this means is that the glucose can still hijack glucose transporters, so it can actually be shuttled into the cells and out of the cells as well. However, it cannot be synthesized by glycolytic enzymes, meaning that it allows it to stay in much higher concentrations in the cell and doesn't break down into lactate. This is important because having that high osmolality inside of the cells really protects the cell. It provides colligative resistance to decreases in cell volume. In other words, it holds on to water within the cell so that it dehydrates less, but it also decreases the freezing point intracellularly as well. So both of those things help to protect so that the cell does not form ice inside, and ice is only formed outside, which is a key feature of partial freezing. We really want to initiate controlled ice formation in the extravascular spaces and maintain an unfrozen fraction inside the cell. The second tool that we use from the wood frog are potent ice nucleators. The wood frog actually has ice nucleators all over its skin. And as ice starts to form outside in the external environment, these initiate ice formation at relatively higher temperatures. That's also critical because what that enables is that when ice forms slowly like that, it sort of creeps through the system. That allows for water to be pulled gradually from the inside of cells to the outside of the cell. And then we can actually control ice formation in that way. And that is another way in which you can protect the intracellular environment from intracellular ice formation. We don't actually use, and in the applications that I'm going to show you, we don't actually use the ice nucleators from the wood frog. We use a different one, but the principle is the same. So these are some of the biomimetic agents that are critical for our partial freezing protocol. The other thing that I want to highlight is that we have sort of a suite of core technologies that we use because, of course, it's not trivial to make mammalian cells that cannot survive this type of stress into ones that can. So we use and employ a whole suite of different technologies to try to understand the process of ice formation, also allow us to tune the amounts and types of crop protectants that are going to be required. And I'm going to take you through a couple of those right now. So the first is we like to model cryopreservation approaches using this capillary on a chip device. This was particularly important because, especially for partial freezing, because we really, as I mentioned, we want to initiate ice formation outside, but maintain intracellular ice formation at 0. So one of the things we did is we actually used, this is actually just a basic PDMS device. We lined it with some extracellular matrix proteins and then some endothelial cells, and you can see the image in the bottom left corner. We can actually fit these into our cryo stage and watch in real time as ice propagates through the system. And we can use this to be able to load certain cryoprotectant agents and understand how ice formation will impact endothelial cells, not just in viability, but also in attachment as well, which are two main and critical issues in partial freezing. So using the system, I'm not going to go too deeply into the data, but using the system, we were able to tune the concentration of some of our cryoprotectant agents like 3-OMG, which is our glucose analog. So using this, we were able to discover that 100 millimolar, so we load these, freeze the whole capillary on a chip model, thaw it, and then look at viability and recovery. And it looked like 100 millimolar of glucose was really the optimal to be able to preserve endothelial attachment, viability, and then later you'll see function as well. It also allowed us to describe how important it was to initiate ice formation at really high temperatures. So if you look at this graph, we have a control here where we froze this capillary on a chip device without any cryoprotectants. Sorry, actually this control is not cryo, so there's no cryopreservation here. When you when you freeze with no cryoprotectant agents, you just remove all of those cells from the system. But if you add ice nucleators and initiate ice at a relatively high temperature, you can actually preserve endothelials without any other cryoprotectant agents, just by that controlled freezing process to the point where it's equal to controls. But as you go down into deeper storage temperatures, you remove that protection. And that's when you have the addition of other cryoprotectant agents like 3-OMG and others that are required to rescue preservation of those endothelial layers. The other way in which we can model partial freezing approaches is using the zebrafish. So as John mentioned in the beginning, where I'm literally freezing whole zebrafish down to -10, thawing and reviving. This is an experiment where we leveraged the really powerful suite of transgenic lines that are available with zebrafish. So this is a zebrafish that expresses M-Cherry and endothelial cells. And we had two groups. So on the left-hand side, this was a zebrafish that was injected with a FITC labeled dextran dye into the circulatory system. This zebrafish larvae was frozen down to -10 degrees Celsius and then thawed. And then what occurred was that you had complete obliteration of all of the cell structures, and in particular the endothelial barrier function, and leaking out of that FITC labeled dextran into the external media. And you can actually collect the external media and quantify this barrier function, or this ability to actually maintain the structure of the endothelial layer within the zebrafish larvae. The second group, which is shown on the right, this was a group where we actually co-injected alongside the FITC labeled dextran other important cryoprotectant agents, in particular that ice nucleator that we've been talking about and the 3-OMG. as well as two other important crab protected agents that are used broadly in crab biology, and that includes glycerol and DMSO in this case. Those were co-injected, and then the same zebrafish were frozen down to minus 10 degrees Celsius and thawed. And you can actually see that after thawing, you have maintenance of that FTSE labeled dextran within the vasculature. And you can actually see circulation in some instances after freezing as well. So you can see how we've actually been able to convert a tropical zebrafish that cannot survive freezing into one that actually can. We haven't been able to show long-term survival with these larvae yet, but nonetheless, quite a difference between freezing without those cryoprotectant agents and freezing with. However, ultimately, we want to use these model systems to be able to scale up to our whole organ models. So what we actually did was we used these lessons from our capillary models and from our zebrafish. So again, being able to tune those different concentrations of CPAs, we tested different loading conditions, the importance of the ice nucleator, and then we applied that into our whole organ system. And what we can see is before we had done all that effort in using those model systems, we had a lot of endothelial cell damage. But after sort of tuning that and learning a lot from those model systems, we were able to scale it up and show a much better outcome in terms of endothelial cell protection after partial freezing. So now I'm going to take you more into our whole mammalian systems. I think I'm probably running a little bit low on time, and I want to leave a couple of moments for questions. So I'll try to go through this quickly. But basically, we have a protocol for whole liver mammalian partial freezing that leverages machine perfusion, the strategic delivery of cryoprotectant agents that use subnomothermic perfusion as a preconditioning step, hypothermic perfusion to deliver some of the cryoprotectant agents that might have a little bit higher toxicity. Then we remove the organ from perfusion, store it in the frozen state, as you can see here, for up to 5 days. Then we remove the organ and then again strategically leverage machine perfusion, both at hypothermic temperatures to remove those cryoprotectant agents, gradually rewarm to subnormothermic temperatures to recover the organ, and then finally bring it up to 37 degrees, so physiological temperatures as it needs to assess the whole organ in the presence of blood to sort of mimic that reperfusion stress upon transplantation. So again, I won't take you too far into the weeds in terms of the data, but basically what we did was we stored these livers at minus 15 degrees Celsius for 1 day, which is shown in pink, and 5 days, which is shown in light pink. And we were excited about the results. We see a couple of important parameters that are used to assess viability during perfusion. For example, resistance does not increase as a function of time, which is an important indicator of organ failure. Our oxygen consumption rates looked great. Our lactate increases were not different from our controls. Most of our livers did produce bile after freezing, however, it was significantly lower from our controls. And then finally, another that we use, which is called energy charge, that was also not significantly different than our controls. That's up to 5 days of storage time. And these are controls that were only stored for the clinical limit in rodents anyways, which is 24 hours. Currently, we haven't transplanted these livers, so we don't know transplant success, but certainly promising data in terms of the perfusion metrics. So I actually had much more to talk about and I went way too far. So I'm actually, oops, I stopped sharing by accident, my apologies. Let me just skip forward, try and share again. I'll skip forward. I missed a lot, but that's okay. To the acknowledgements. I do want to thank many of the contributors to this research, of course. I'm the lucky one who gets here to talk about all this exciting research, but there's so many people who are behind the scenes doing all the hard work. Includes Luciana Manuela, who are leading a lot of the heart research. Casey, who has been pivotal in really all of the data that I showed you today. Rainier, who led all of the supercooling experiments. A wonderful mentorship team, Mehmet and Korkut, and many of the transplant surgeons, of course, that we work with, and many of the patients and organ donors who have donated organs for research, and all of our research support as well, including National Science Foundation.